Thank you very much, Nikolai, for that very kind introduction. I just want to say also thanks to all the organizers for putting together this wonderful seminar series, and I'm so happy to be a small part of it. It's a really wonderful opportunity to share our work in the laboratories of Ben Ebert and Marcella Moss, really at the interface of targeted protein degradation, synthetic biology, and cellular immunotherapy. And I just want to start by acknowledging everyone who has contributed to the work from these two wonderful labs. And most of all, it's just such a wonderful thing that I have not one, but two really superlative world-class postdoc mentors, and I really do not take that for granted. So thanks to everyone in these two wonderful groups. CAR T cells are a genetically engineered cell therapy in which, in general, a patient's own T cells are collected, stimulated, expanded, and then transduced with a chimeric antigen receptor before finally being returned to the patient. The chimeric antigen receptor over here on the right in green binds a cell surface tumor antigen and is a single pass transmembrane receptor. The binding event activates signaling domains that in a lot of ways approximate normal T cell activation, which results in tumor cell killing cytokine release as well as proliferation of those T cells. And the experience of the first child to receive this form of therapy, Emily Whitehead in 2012, is very illuminating. So on the one hand, CAR T cells are a bona fide medical breakthrough. They've transformed otherwise dismal outlook for patients with a number of relapse refractory B-cell malignancies. But on the other hand, they are hard to take, they're unpredictable, and they're capable of life-threatening toxicity. As some evidence to that, in Emily Whitehead's case, a few days after this therapy was infused, she had a T-cell hyperactivation syndrome called cytokine release syndrome, which sent her to the ICU, was placed on a ventilator with a 105-degree fever, and really only through incredible perseverance and truly elite medical care was she able to survive this cell therapy. And so I think we can agree that the promise could not be more meaningful, but the process has to be better. And we as a field can't become sensitized to asking so much of the people who are taking these therapies. So the root cause of these toxicities is that today's CAR T cells traverse what can really be best described as a ballistic arc that is very hard to control once these cells are infused. Over the first few weeks, CAR T cell therapy can expand 1,000 to 10,000 fold within a patient. And T cell hyperactivation syndromes are unsurprisingly quite common when you have many billions of T cells that are simultaneously interacting with and activating against many billions of tumor cells. And so it really doesn't take a lot of imagination to envision a safer and a more regulated paradigm in which the cell therapy can be precisely turned down to prevent especially early severe toxicity, or up in the case of disease persistence, and ideally having a system that can be routinely tuned from day to day as clinical situations require. And this tunable, titratable dosing model is really a basic expectation of the majority of medicine, and certainly cancer therapy, whether it's chemotherapy or radiotherapy, against which the uncontrolled pharmacokinetics of today's cell therapies really stand out in very sharp relief. These achievements are decades in the making, starting at least in this graphic in the 90s with primary T cell engineering, and in the aughts with preclinical development of eventual CAR T cell therapies. And we have over a decade of experience culminating in the first clinical approvals in 2017. Another way of saying this is that the field has known about the the inherent drawbacks and limitations of these fully autonomous, uncontrolled cell therapies for over a decade. So I think it's fair to ask, where are the next generation designs that can overcome these fundamental limitations? And so I think that's a complicated question with multiple stakeholders, but part of the answer lies in the synthetic biology that's been available to make this next generation. So CARs have been the favorite setting to develop and to demonstrate new chemical and synthetic biology techniques to both engineer autonomous behaviors as well as user control, and really just a very partial list of the incredible diversity and strength of control systems are listed here. But despite this enormous investment and many beautiful scientific works, a very small number of these technologies have been able to go forward into clinical testing, and really only a handful of companies are at the very beginning of clinical testing of more controllable cell-based therapies. So why is that? One major issue is that cell therapy imposes very narrow design constraints on the systems that are used within them. And so clinical suitability in this setting really means all of only using human sequences to avoid immunologic rejection of cells containing these systems. And any controller drugs should be all of clinically approved, well-tolerated, and non-immunosuppressive. And unfortunately, this undermines many of the best characterized synthetic biology systems that originated in model systems or have been built around molecules like rapamycin and related molecules that are very potent T-cell killers in most settings. And so this leads us finally to targeted protein degradation. So with this unmet need in mind, we set out, as Mikolai mentioned, several years ago to start to try and build a toolkit of molecular switches and more complex regulatory strategies around the chemical biology of thalidomide analogs. And the opportunity comes out of the discovery over the past several years of the remarkable mechanism of action of thalidomide analogs as targeted protein degrader drugs, acting as molecular glue between CRL4-CRBN ubiquitin ligase and a growing list of known neosubstrate proteins that are subsequently ubiquitinated and proteasomally degraded. And it is the degradation of disease-relevant neosubstrate proteins such as IKZF1 and 3 in multiple myeloma, or casein kinase 1 alpha and MDS that explains the therapeutic, anti-cancer, clinically approved mechanisms of action of these drugs, in particular lenalidomide and pomalidomide, which are in mainstream daily practice for a number of hematologic malignancies. So with this as a background, we have set out to engineer molecular switches gated by lenalidomide, and ideally a whole toolkit of these, and with them to generate switch controllable cell therapies using these technologies. So with that, I'll present some of the data from this work, our collaborative work that was published yesterday. And this really started with the thought that because no evolutionary process has really been definitively proven to have optimized these complexes mediated by fully synthetic drugs, we are optimistic that higher efficiency degrons, what we've been calling super degrons, could be engineered from the starting point. And as an engineering framework, C2H2 zinc fingers are stable, they're small, they're modular, and importantly, multiple of them have been found to engage a portion of cereblon with a conserved structural degron motif. And you can see two superimposed zinc fingers binding upon this this cereblon and pomalidomide composite surface. So we set out to generate diversity within this starting sequence space by taking 22 known C2H2 zinc finger degrons and generating a hybrid zinc finger library in which every possible N and C terminal combination of these sequences was generated and then encoded into a pooled lentiviral protein degradation reporter assay in which the zinc fingers were fused in frame with GFP so that their protein abundance could be easily tracked. This lentiviral pool was transduced into GERCAT human T cell line. And then these cells were then exposed to various drug conditions, sorted by FACS, specifically for the GFP lows that in the drug treated conditions would be indicative of or enriched for sequences that had been efficiently degraded, and then used next generation sequencing to deconvolve the relative abundance of each element. And so presenting some of the findings from this functional genomics approach, here I'm presenting the data from these screens ranked by the overall enrichment of N and C termini. And for each particular sequence, their significance is denoted by the size of the circle and their fold enrichment by the color. And this is comparing, again, the GFP low gate with lenalidomide treatment versus vehicle control. So the sequences that are enriched in this comparison are candidates for being efficiently degraded in a drug-dependent manner, thereby being more prevalent in this gate. So what you can see is that the vast majority of the strongest hits were really in this subset of this graph, where a fairly promiscuous set of combinations of a half dozen N and C termini resulted in the strongest hits in this experiment. And all of these, except for one, are novel hybrid sequences. The only native zinc finger is IKZF3, which is here. So the preferred N-terminal beta hairpins converged on a similar sequence at a couple of residues that are known from prior structural work to be direct sidechain drug interactions. But as you can see, the rest of these sequences were otherwise molecularly diverse. So I think there's still quite a bit to learn through additional diversification of sequences and this type of functional genomics approach. To carry forward with particular hits from this screen, we, in a single construct validation format, exposed the control IKZF3 or a number of these hybrids again as reporter constructs to varying concentrations of lenalidomide or other drugs to calculate the half maximal degradation concentration. And these hits were found to have DC50s that were overall 2 to 6 times lower than the IKZF3 control. And among fairly equivalent options, we chose to go forward with a hybrid of ZFP91 and IKZF3 for work in primary T cells. So these were 23 amino acid minimal C2H2 zinc fingers, and then we added a little bit of additional sequence on either end to go forward with 60 amino acid degron tags. So the first design that we worked with was to generate drug off-switch degradable CARs that in the absence of controller drug would be expressed and would function like normal CAR T cells, but once a thalidomide analog was added, that would induce degradation of the receptor, rendering those T cells inert in a process that could be reversed upon metabolism and excretion of the drug, reactivating those T cells. So we started, first of all, by comparing the efficiency of degradation of an IKZF3 based degradable CAR with this ZFP91 IKZF3 super degron based CAR, and did so in a number of manners. and this Western blot is representative of those efforts. And what we found was efficient degradation of the IKZF3 based degradable CAR at around 10 nanomolar lenalidomide. Whereas for the super degron tagged CAR, we saw efficient degradation about an order of magnitude below that, around 1 nanomolar. For a bit of context, the peak plasma concentration in myeloma patients receiving a starting dose, lenalidomide dose is approaching 2 micromolar. And so these are certainly concentrations that got us excited about the possibility of using this system to eventually be able to control protein abundance in a clinical setting. The other thing that was encouraging was that we could see about a 2 order of magnitude separation in the dose required to deplete the super degron tagged CAR versus endogenous substrates like IKZF3, again suggesting that a subtherapeutic concentration of lenalidomide or pomalidomide could be used to control these sequences. So based on the difference in efficiency of degradation of these 2 CARs, we focused for the most part on the super degron tagged CAR on the right. Moving forward into primary T cell models. So what I'm showing you here is a live cell imaging time course experiment co-culturing GFP positive tumor cells with mCherry positive CAR T cells. And you can very satisfyingly see the very potent anti-tumor effect in this in vitro culture assay over 24 hours. And the result of this is really consistent with the hypothesis that the addition of the super degron tag did not impair the anti-tumor function of the degradable CAR T cells. And then to test the effects of lenalidomide on either CAR constructs, we then performed the same experiment in the presence of 1 micromolar lenalidomide. And as you can see again over the course of this time course, on the left-hand side, the conventional CAR is essentially unaffected by lenalidomide treatment, whereas on the right, even though you can see, if you look carefully, mCherry positive cells that are in this assay and interacting with tumor cells, the killing function is essentially completely inhibited. And so we tested the effects of lenalidomide in more standard tumor cell killing assays as well. So this is an example of that, where on the y-axis is the specific lysis of NALM6B ALL tumor cells across a range of effector target ratios. So tumor and T cells are co-cultured together overnight, and then the lysis of tumor cells is evaluated after that. And as you can see, comparing a conventional CAR in gray, the degradable CAR in blue, or the untransduced T cells in black, there is efficient constant or ratio-dependent killing effect that is entirely inhibited with 100 nanomolar lenalidomide. And this complete control also extended to cytokine release and T cell proliferation as well, although I won't show that data. We examined the kinetics of off-switch CAR depletion after the addition of lenalidomide, first in vitro. And as you can see, the addition of lenalidomide results in the establishment of a new steady state of much lower surface CAR abundance in about 1 to 2 hours after drug addition. Whereas when we examined the dynamics of CAR resynthesis after washout of lenalidomide, it took a little bit longer, really as expected, for the degradable CAR to be re-expressed and then to accumulate on the cell surface, still in a matter of hours. So all of this being consistent with the hypothesis that we could control CAR abundance in potentially clinically relevant timescales. We then extended this analysis of the CAR abundance to an in vivo model. And so we engineered CAR reporter constructs that were fused in frame with luciferase with either a degradable CAR or a negative control. And then this construct was introduced into, again, the JERCAT human T cell line, and JERCAT cells expressing these reporters were engrafted into immunodeficient mice and allowed over the course of several weeks to expand to the degree that the CAR luminescence could be readily detected by whole body bioluminescence. Then we gave 1 dose of pomalidomide orally and tracked the CAR abundance 6 hours after as well as 24 hours after. And as you can see, 6 hours after, there is a significant greater than 5-fold decrease of CAR luminescence, specifically for the degradable CAR that was fully recovered by 24 hours, again demonstrating reversible control of abundance of the tagged protein in a potentially clinically relevant timescale. So finally, we tested whether CAR degradation could be used to suppress inflammatory cytokine release, the functional effect that is really the major driver of CAR T-cell hyperactivation toxicities. And so in this model, we engrafted immunodeficient mice with, again, the same B-ALL human tumor line. And then a week later, adoptively transferred CAR T-cells to treat these mice. In previous experiments, we had determined that it takes a few days for there to be a peak in human cytokine release into the blood. And so we treated these mice for 3 days with a couple of different drug schedules with pomalidomide. And as you can see, measuring blood cytokines after that. And as you can see, multiple inflammatory cytokines are significantly suppressed by either daily or twice daily pomalidomide treatment. In separate experiments, we determined that this short 3-day drug dose and schedule could be used without inhibiting the long-term anti-tumor effect of these cells. So in summary, this pair of experiments shows that we could pause the release of an important inflammatory mediator from these cells at the peak of their production while still allowing for a long-term anti-tumor effect. So to summarize the first part of my talk, we were really excited that really quite a limited small functional genomic approach allowed us to engineer higher efficiency super degron zinc finger tags. And with these tools, we constructed lenalidomide off-switch degradable CARs that could be efficiently degraded by sub-therapeutic lenalidomide concentrations and could be functionally controlled both in vitro and in vivo. And I think it's worth pointing out that just a few weeks ago, a really excellent paper from a group at Novartis just down the road published a very complementary and similar set of data, in this case using, again, an imide-inducible degron to regulate chimeric antigen receptors. And in their case, they used a portion of IKZF3, including this critical zinc finger 2. And then very similar between the two papers, we're able to show a drug off-switch design. And there are certainly important contrasts between the two papers, but I think one of the most important things is that this provides really immediate cross-validation between the two independent groups, and I'm really happy to see it. It's really terrific work. We were, back in 2017 as well, very interested in trying to generate multiple strategies and potentially a lenalidomide on switch. And the strategy that we took to do so was to see if we could generate a lenalidomide inducible dimerization system around which we could build a drug on switch receptor. And what allowed us to get started on this project was really the foundational seminal description of the structural basis of how this chemical biology works. And what stood out to us from the structure of CRL4-CRBN in complex with thalidomide is that the substrate adapter cereblon really appears to be a bifunctional molecule in a way that could potentially be separable. So the drug and neo-substrate binding domains are essentially entirely separate from the alpha helical bundle at the bottom of this representation that mediates all the important contacts with DDB1 and recruits the rest of the ubiquitin ligase. And so we hypothesized that we could generate variants of cereblon that that would lack this recruitment of the ubiquitin ligase, so that you could have a drug-induced heterodimerization event between one of these variants of cereblon and a zinc finger without recruiting the ubiquitin ligase and degradation of that zinc finger. And so to get started with this approach, we generated a number of cereblon deletion variants that lacked this key alpha helical bundle that binds to DDB1, and then used a bioluminescence resonance energy transfer system for in-cell measurement of drug-induced dimerization. And so what we found is in the positive controls, a drug-dependent Brett signal in black, and although weaker, also a drug-induced heterodimerization Brett signal with one of these variants, deletion 3, which only takes out this alpha helical bundle in these Brett experiments. So our next step was to start to assemble these polypeptide elements into the framework of a split chimeric antigen receptor. And we did this by placing cereblon into this portion of cereblon into a cell surface protein, as well as the same with a portion of IKZF3 containing the critical zinc finger. And this was done with a number of different cell surface tethering and signaling domains. And the take-home message is really across the various domain options that we tried. In every case, as you would expect, restricting these elements to the two-dimensional cell surface limited spatial environment increased the Brett signal for both the tonic Brett signal as well as drug-induced signal. And then we were able to further enhance our drug-induced Brett signal by protecting the zinc finger containing component of this receptor from endogenous CRL4-CRBN, which should be able to continue to bind, ubiquitinate, and degrade this cell surface protein, but by making K0 mutations, in this case, all lysine to arginine substitutions in the intracellular domains of this protein, we were able to enhance, as I mentioned, the efficiency of the system. So what we had built, to summarize, was a split receptor where the key signaling domains are on a separate protein from the antigen binding domain. So only in the presence of lenalidomide is the split receptor dimerized such that it can activate upon antigen binding. And to start to test this potential on-switch control, we used, first of all, a Jurkat cell line model of T-cell activation, again, using co-cultures of tumor and Jurkat T-cells expressing these split receptors, comparing a conventional CAR to a split receptor using the IKZF3 derived zinc fingers, as well as also a split receptor including the ZFP91 IKZF3 hybrid that we had characterized earlier. And as you can see, there is most efficient tunable on-switch control of the activation of these cells. CD69 is an early activation marker following T-cell activation with the hybrid zinc finger derived split receptor. And then to test our ability to control effector functions in primary T cells, we again performed a NALM-6 tumor killing assay across a range of lenalidomide concentrations and saw tunable on-switch killing as one increases the lenalidomide concentration for the split CAR in comparison to the conventional or CAR or untransduced T cells. And in a final in vitro experiment showed that the proliferation of these cells also required both the presence of antigen as well as lenalidomide for the split CAR. And so in this assay, we use a DNA intercalating dye for just a pulse to label each cell's DNA, and then over the next several days, expose those T cells to antigen positive cells and allow them to proliferate. So the untransduced T cells really are not activated in this assay, and you can see undiluted crystal violet staining, but the conventional CARs are proliferating with or without lenalidomide in this assay. There's a different pattern with the split CAR in that in the absence of lenalidomide, there's really very little dilution of the cell trace violet signal without lenalidomide, but with lenalidomide, there is much more significant proliferation. And then finally, to test the switch control of anti-tumor activity in vivo, we used a model in which, again, immunodeficient mice were engrafted with tumor cells and then treated with CAR T cells. Following that, pomalidomide was given daily for the next 11 days. And what you can see on, in the center here is that with the untransduced T cells, you can see with or without pomalidomide, the unopposed expansion of the tumor cells over this time course. With the split CAR, you can see a pomalidomide-dependent anti-tumor effect that almost entirely clears these mice in this experiment of the tumor cells. But since there was a little bit of residual tumor activity, we could then ask whether those tumor cells were still being suppressed by the split CAR once we withheld the controller drug. And this allowed us to see that the on-switch split CAR is also a reversible system. And so after the drug treatment period, the tumor started to proliferate again in an unopposed way. So to start to wrap up, we have, in addition to the super degron engineering and our off-switch controls, we've also built this lenalidomide-induced dimerization system, and with them on-switch control of a great many of the behaviors of CAR T cells that we would want to have full control over. So I just want to spend a few minutes as we wrap up on some, I think, very exciting directions that we have started working on and will continue to work on over the next few years. One is this realm of tool development, and being very encouraged by the fact that we were able to identify these higher efficiency zinc finger tags with, again, just a very, very small functional genomic screen. And there's certainly such a diversity of potential engineering approaches to continue to engineer proteins around this chemical biology for higher and higher efficiency degrons, but also to generate even more novel forms of chemical genetic regulation. And so it's been really exciting to begin to work on these ideas, in particular with Eric Fisher's lab. And I think, I guess I have a hunch that this area might be something that might be very interesting to potentially a number of people who are participating in this symposium. And I think that it would be a really wonderful thing if we could do this in as collaborative a way as possible, because I think that there is just so much creativity in terms of the engineering approaches and the downstream goals of this type of work. I think this would benefit from quite a bit of broader groupthink. With the tools that have already been built within this space, we are very excited to explore other opportunities for chemical biology for learning by design as well as biotechnology applications. And it's been really exciting to work with Amit Chaudhry's group on a completely CAR T cell unrelated project that gets at the beginning of the generalizability of these systems. But certainly these systems and recruitment to CRL for CRBN in the has been well validated for nuclear proteins, cytoplasmic proteins, single-pass transmembrane proteins, and whether this or other related systems can be extended to even greater expanses of protein populations is something that we'll be very excited to explore. My own personal interest is trying to really hack and hardwire control over what are some of the most interesting control points of immune cells and the potential novel design space for immune cell therapies that could be harnessed if we have control over the signal transduction and machinery governing cell fate decisions. That could really open up not just safety switches, but cell therapies that have higher efficacy based on these modifications, and also hopefully very tightly, very tight control of those new elements. I think the most exciting thing is really what got us started on this project in the first place, which is the hope and the desire to really try and push these ideas forward directly into clinical translation. And one can imagine with both the off and the on switch, the many challenges that are ahead, but also the opportunities to hopefully make a meaningful clinical impact. And so for the off switch CARs, one very clear use case would be to use them as a reversible safety switch, as a short-term pausing in situations or in disease entities where CAR T cells have already been shown to have preliminarily have efficacy, but have been difficult to pursue and to develop because of toxicity questions such as cytokine release syndrome. So a setting like adult B acute lymphoblastic leukemia might be an ideal setting to try and test these switches in bona fide investigational CAR T cell therapies. And then with the on switch, one might imagine a different landscape of initial use cases. One might be unproven targets, especially for solid tumors, where the target antigens that are being used in CAR T cell therapies are not 100% exclusive to the tumor cells and completely absent from normal tissues. So having a system whose basal state is not active might be the safest and most prudent way to go after a new target that might have toxicity issues. And then another potentially very attractive approach would be to go into the hematologic malignancies where lenalidomide and pomalidomide are such active and important anti-cancer agents, and have a synergistic combined effect of both the CAR T cells and the controller drug that's being used to regulate them. So with that, I just want to one more time acknowledge everyone who has contributed to this work, and thank you all for being here.